first of all, congratulations on your winning Keio Medical Science Prize. And how do you feel in winning this prize?
Well, I've known about the prize for a long time, and I've seen the scientists who win the prize are very, very good scientists, both from Japan and internationally. So it's a great honor to be a winner. Yeah, sure.
Yeah. Okay. And also, I'd like to ask about wind signal pathway and what makes you interested in wind signal pathway?
Yeah, that was totally by chance. So I started as an immunologist, did a PhD, did a postdoc in the US at Harvard and Boston. I came back to Holland, became professor in immunology, and we wanted to clone transcription factors, genes that control how stem cells become T lymphocytes. And when we cloned a gene called TCF1, and we didn't know exactly what it did, but it turned out TCF1 means T cell factor 1 for T lymphocyte 1. But then it turned out that it was the endpoint of the Wnt signal transduction pathway, which we were not studying, but we had a key piece of the puzzle. So I then turned my lab from an immunology lab into a Wnt pathway lab. But it was not decided by me. It was decided by fate.
So it's kind of your luck.
It was but just luck. Yeah.
Okay. And why did you choose LGR5, and why could you believe LG5 is an answer?
Yeah. So what we discovered after we found the Wnt pathway, we made mice that lacked certain genes from the Wnt pathway, and one of the mice that we made had no crypts in the gut, had no stem cells in the gut, and so we then solved how Wnt controls genes in the gut, and we found both you need Wnt for normal renewal in the gut, but you also cause cancer in the gut, colon cancer, when you get mutations in the pathway. So we combined those two. We had a long list of genes that were controlled by the process, about 200, and just one by one we looked through the genes, and we tried in various ways, and a lot of the ways did not work. So this was Nick Barker that mostly did it. So he finally found LGR5. That was very, very hard work. It took about 6-7 years before we found, in that list of 200, we found the right gene. Yeah.
So you tested each gene one by one?
Yeah, different ways.
Different ways.
Where is it expressed? If we knock it out, what happens in a mouse? With antibodies, see where the protein was.
So in various ways?
Yeah. Yeah, depending on what kind of gene was on it.
That's very hard.
Yeah, that was hard work, and it took a long time.
And do you have any comments on Dr. Toshiko Sato in this laboratory?
Yeah, so Toshiko Sato joined the lab, I guess, in 2007, 2008, right when we had discovered the stem cells of the gut with LGR5 that we just mentioned. And so we call him Toshi. So Toshi had in the lab had already had some experience with trying to grow gut stem cells, crypts in 2D on plastic. So so when he joined my lab, we decided, well, maybe we should try to do this in 3D. And then we knew quite well what kind of growth factors should be important that we should add. And so that's what Toshi did, and worked quite soon. It took him maybe half a year to a year.
Really?
And I should also say that then it was believed that was not possible. It was people thought that it was impossible to grow normal cells from a body. So we knew, of course, from the embryonic stem cells and from Shinya Yamanaka's iPS cells that you can grow them for a long time. But still, almost everybody thought if you take skin or you take gut or liver and you put it in a dish, they live for a few days, the cells, but then they will die. So it was quite courageous for Toshi to take up this project, because other people in the lab said, don't do it because it's not going to be successful. So he's done this. It was a big breakthrough, I think. And since then, when he was still in my lab, he had several others. So when he could grow the gut, he could grow other organs. And then when he went back to Tokyo, he also has done great work. And actually, I just heard from him today that he published a paper in Nature today.
Oh, really?
Right.
Wow, nice.
So my questions are, Why and how did you decide to dedicate your life in the basic research rather than in the clinical medicine?
Yeah, that was when I was 4 years old.
4 years
No,
Right.
so I always knew as a young child that I wanted to be a scientist.
Oh.
And I was then interested in animals and shells and minerals and things like that. And I've read a lot of books about discoveries so in Africa and in the North and South Pole, but also scientific discoveries. So I always wanted to be a scientist. But then I did biology at the university and also medical school. Then for a while I thought, well, it's a very nice life as a doctor. It's very interesting, very social. So I had a pediatric training position. They then told me, why don't you, because you have this double background, why don't you first start a little bit of research and then you go into pediatrics training. So in that year I decided, well, maybe it's not so nice in a lab, maybe it's nicer to work in a hospital from day to day, but I'm really a scientist. So that's when I decided, it was a difficult decision, that I should really be a scientist. And so I feel that it's very, very difficult to be both. So doctors who want to be scientists is very, because you have to be good in two things.
Yeah.
So I decided I just go for science. Hopefully it works, hopefully I'm lucky. Otherwise I would have been a doctor, but also I would have been only a doctor. I would never have combined it. So if I look back, it was a good decision, but it was quite risky.
Right. How is it difficult to do both? I mean, is it by time or?
Well, they're both difficult jobs, difficult professions. And well, if you do one thing, you have more time, you have more concentration, more focus than if you do both things. So the fact that I studied both biology and medical school has really helped, because although I was a basic scientist for a long time, I speak the language of doctors, because I was at medical school, I have an MD degree, that really, I always worked in a hospital the first 10, 15 years. So that really helped in the end, now that we all of a sudden can work on diseases with these mini organs that Toshiko Sato developed, now we talk to many doctors, and it makes it a lot easier for me, because I know the diseases, I know many drugs, to be able to communicate and collaborate with the clinic.
Thank you. And have you had any hard time in your research life? I'm pretty sure you have. And how did you overcome those situations?
Yeah, so there are always times in research where things don't move, when you have the wrong ideas or when the technology does not yet exist. And so we have had several times where it went very slow. So sometimes, in my experience, science always goes in, it goes fast, it stops, it's hard work, it goes fast again, it stops, it's hard work. And so the first time the hard work was when we just cloned TCF1 in the early 90s. And we didn't know what it was doing. And it took 5 years. So several young people worked very hard until we discovered that it was a part of the Wind pathway. So then we had a few good papers, and we became a little famous. Then we wanted to know what they control, and then the technology didn't exist. Microarraying was invented in 2000, so we had to wait 3-4 years before we could use that. Now, we didn't know that it was going to be developed, so we tried all sorts of things, didn't work. Then microarraying was developed by Pat Brown in California. We collaborated with him, and then we got a list of 200 genes. Then we again had to work for 6 years or so to go through all these 200, find LDR5, and from then on it's been easier, because then we had the stem cells, we had many different stem cells. Then Toshiko Sato developed the mini organ, the organoid technology. We could make many different organoids. So since then, also the lab is much bigger now. I get very good people in my lab now, young people, very creative. So the past 10 years I've been extremely busy, but it's no longer very difficult. But there were 2 or 3 periods that I described where we were not moving, and we never knew, are we in the wrong direction, or are we going in the right direction, but we need to work harder. And that's a very difficult decision, because you only know when you look back if you took the right decision.
Then what drives you and motivates you through the difficult time?
Well, it's really the curiosity. So I always tell my people, science is a great profession, to be a scientist, but you have to realize it's very hard work, and it comes with a lot of frustration, because you try many things that don't work, and sometimes the thing works. So if you get enough satisfaction from a breakthrough every once in a while, and if you can deal with all the other frustration, I think then it's a good profession.
Right. Okay. So, when and how do you come up with these new research ideas? Is there any specific moments that?
Yeah, there's several. I don't think there's a consistent pattern, but so I go a lot to meetings. I don't read papers so much, but I basically go to meetings and I talk to the other speakers. So and then you get much richer information. You hear, well, that paper, we don't know whether it's really true. That paper is a fantastic technology. You should like to. When CRISPR was developed, we immediately knew when we could apply it in our lab. So that's one thing, and then discussing with colleagues around the world. Often that's when ideas come up. Or I listen to a lecture, and then your mind starts wandering. You don't listen so well, and all of a sudden you see a slide or you hear a word, and then all of a sudden it connects in your brain, and you get a good idea. And in other cases is by just discussing in the lab. So if something doesn't work, who has a good idea? What shall we do? And then try to get different ideas and then test them out. I think those are so it's usually interaction with other people. It's not when I can sit in a chair on my own, think very hard, and then there's no answer. I never get an answer.
Okay, so you spend time on your networking and connecting with others.
It's mainly talking to people in the lab, talking to colleagues around the world, exchanging ideas. What do you think? Do you have a good idea? Yeah.
Right. Thank you so much.
So, as a principal investigator, what was the most important thing to run your own lab?
Yeah, what I have found very important, and also what my mentor in Holland, when I was a PhD student, what he found very important is that as a scientist, you have to have trust. You have to have trust in yourself, be confident enough, be courageous. Like Toshiko Sato was very courageous when he picked a project that everybody else told him don't do it because it's not going to work. So you have to have confidence that you can do it, but maybe even more important, you have to trust other people. So trust the people in the lab, but trust all the scientists around the world. So I see a lot of scientists who are maybe a little bit afraid. They don't trust others, so they keep things secret. They don't share information. They're afraid that they get scooped or that somebody else will steal their ideas. I think that's very rare. So in my lab, I always tell the people, collaborate with everyone who wants to collaborate, be open, tell them everything that we know, share that with them. Every reagent that we have made, mouse, we always immediately share with other labs. And that, first of all, is very nice because you make friends and you can communicate but also it builds a network that maybe 3 years later you all of a sudden need something, and then I can just call or I email and said, remember we met there and there and there, and we sent you this antibody and we talked, and can you now help me? And then these people, because I trusted them 3 years ago, they will trust me. And so I think that is very important. It makes science much more rewarding. If you're not even suspicious and you're not paranoid, but you trust people, then every day is much nicer.
Well, didn't you have a fear that you were doing wrong thing when you're doing research?
Yeah, well, most of the things we do are wrong, because you just test many things. So I think you should not be afraid that you do something wrong, because even if it's wrong, you'll learn something. You always, maybe you see something. What is most important is that that there is a point when you're going in the wrong way. It's like when you're walking and you walk in the wrong way. Sooner or later you realize this is not where I should be. And then it's a good moment that you say, okay, this is wrong, I go back. That is the most important point. So don't continue when it doesn't work, right? It gets worse and worse. So it's more important. So you can start many projects, but also you should stop them if it looks like you're going the wrong way. And that is a very difficult decision. It's much easier to start a project than to stop a project. And I think my lab has always been very good in trying many things and then throw most away and then take the one that looks good and then go ahead. So that has been our strategy usually.
Thank you. So what was the hardest thing while organizing your laboratory as a principal investigator?
Yeah, so I think the hardest part is really the first few years when you have nothing in your hands. You come out of your postdoc and PhD postdoc, essentially you work on your own. You collaborate a little bit in the lab, but the only thing you do is your experiments. And so when you're a postdoc, you think that's the hardest job in the world. But actually the next step, so then from being only in control of yourself and controlling your own, planning your own experiments, when you become a PI, you all of a sudden have to do many more things. You have to have a strategy for more than one person. You have to hire people. You have to get projects going. You have to troubleshoot, support when things don't go very well. No, you have to decide stop a project. Very hard, as I already said. But also you have to build a network, and nobody knows you. You have not made any discoveries yet. You have to get grants, you have to give talks, you have to, all these things, they all come at the same time. And usually it's also at the time that you're building your young family. So it's usually you're in your 30s, so you probably have bought a house, you might have, get some children and got married, and so that all comes at the same time. And once you get more experienced, you know better how to deal with how to write a paper, how to write a grant. Should I give a talk or should I send somebody else? But in the beginning, you have no experience. Nobody knows you, and somehow you have to make it through that hurdle.
And
luckily, it's a long time ago for me.
And we read your career, and we found that you almost stayed in Dutch as your research career. And you went to US while your postdoc period. So I want to ask you, why did you stay Dutch, not in the US?
Yeah, so I was in the US for 4 years with my wife, and I think we wanted to raise our children in Holland, and they were born after we returned. Also, I think you don't need to be in the US to do the best science. I mean, Japan has a good example, and many good Japanese scientists who always worked in Japan. In Holland, the same thing. There's some fantastic scientists who never left Holland. And so, I think my experience is that when you have the equipment you need and you have motivated young people, you can compete with any lab in the world. So you don't need to be at a top university in the US to do good science. And I think in Japan, now also in China, many places in Europe are as good as the good places in the United States. I go a lot to the US. Of course, it's still the biggest science world. They do most medical science, they win most prizes, they make most discoveries. But the level of individual institutes here or in Europe is the same as the best institutes in the US.
That's a very confident comment, because young scientists like us wants to go to US because it seems like a dream country of the science.
Well, there is something about the US science, it's extremely competitive. And our system and also the Japanese system is different. So in our system, quite a lot of the money that comes from government goes to the institutions. And then there is money that goes to the granting agencies. So in Europe and in Asia, often you already have people, labs, equipment to work. And then on top of that, you start writing grants. In the US, in most universities, there is nothing. So you have to write grants, and if you don't have grants, you don't even have a salary yourself. So that makes it very, very competitive. And I must say, I felt that the Dutch system, where there was always some money in my lab to pay for my salary, to pay for a few more people, and these are the people that did these very risky things, because they didn't have to be successful in 3 or 4 years. Mark van der Wetering was one of them, so he could invest 3, 4, 5 years. And then make a discovery. So that's essentially how we could bridge those periods that we discussed, where we didn't make discoveries for 5 or 6 years. And because I always know, well, there is always money. Even if we don't make discoveries for 5 years, we write some papers, but they're not breakthroughs. But they could actually invest long term. In the United States, this would be difficult, because if you have not made discoveries in 4 or 5 years, there's no grant, and then it's the end of the lab. So there it's more risky to take risky projects in the US than it is in Japan or in Europe.
Thank you so much.
No.
So as a last question, could you advise students who aim to become a physician scientist?
Yeah, so I think a physician scientist has to realize that you're doing two things, and you want to be very good at two different things, which both are difficult. So that's one challenge. And I guess most physician scientists, the ones that do best, are the ones that, when they keep on doing clinical work, that they do research that is more translational. So they do research that involves patients. Extremely important, because when people like me make a basic discovery, the next step has to be in the clinic. So it's exceedingly important that there are people in the clinic who understand science and can do clinical research. So I think that's one way by which an MD can be an extremely good scientist, by being a clinical researcher and have an open eye for people like me who are basic scientists. The other way is if you are an MD and you want to do more basic science, you probably have to limit your clinical activities a lot. So maybe just focus on one rare disease, be the expert in that one rare disease, treat patients with that disease, but spend most of each week on your research. Because if it would be 2 days research, 3 days in the clinic, then you cannot compete with people who do either of the two full time. Yeah. But even the people that I know that are clinicians and have very limited clinical duties, in the US you have many of these people. Look at Harvard, at Stanford. They maybe see patients once a week for 1 day, or maybe 1 month in a year, and the rest they do science. They still benefit a lot from their medical training. They understand diseases very well. They know if they need to collaborate with the clinic, they know exactly how to do that. So both types of research are very, very important.
Thank you. So that's the end of our interview. So thank you so much for Professor Hans Clevers.
Thank you very much. Very good interview.
Thank you.
Thank you.
Thank you so much.
Thank you very much.
Thank you so much.
Thank you very much.